mathematics with problems from science, technology, and society. So I think some of you may be interested in the program that's coming up in the fall, which is on genomics. We're hosting 2 workshops that might be of interest, one on microarray and proteomics, and another one is the Recom workshop on comparative genomics. If you visit our website, you'll see some details about that workshop and how to register and so on. So the next public lecture is on June 4th, and it's by Charles Peskin. There's some information up there on the posters up there on the top of the room. It has an attractive title called The Secrets of the Heart Revealed by Mathematics and Computer Simulations. So now it's my pleasure to introduce today's public lecturer. Eugene Myers was formerly Vice President of Informatics Research at Celera Genomics. I think he's most well known for one of the persons who worked really hard in sequencing the human genome using a shotgun technique that he'll talk about today. He was awarded the IEEE Third Millennium Achievement Award in 2000, the Newcomb Cleveland Best Paper in Science Award in 2001, the ACM Kanellakis Prize in 2002. He was voted the most influential in bioinformatics in 2001 by the Genome Technology magazine, and was elected to the National Academy of Engineers in 2003. His research interests include designs of algorithm, pattern matching, computer graphics, computational molecular biology. He's developed a bunch of tools that I think some of you may have used. BLAST is one of them, Factory, NREP, Mac Molecule, and PC Molecule. He's currently professor of computer science at the University of California, Berkeley. So, Eugene?
Thank you. It's a pleasure to be here. So you may have, thank you. We'll see if you feel that way at the end. So as you may have noticed, the lamp needs replacing, and you're going to notice that throughout the talk, and so we'll try to work around it. The titles will come through there, but I'll go around them. So I wanted to kind of start in the past, namely in 1981. When a little virus by the name of Lambda, which was about 50,000 nucleotides long.
The volume a little bit up.
The volume needs to go up? Is that this? How's that?
Good.
Better? Okay. All right, great. Thank you. So Lambda was 50,000 nucleotides long. It was sequenced by the shotgun method by Sanger and his team. And I recently read this paper. I'm preparing a review article, and I was really impressed with the paper. It had many concepts, including end sequencing, the use of DITP, and really was a landmark paper. Basically, the basic idea is that you had a piece of DNA, say those yellow lines represent the virus Lambda that's about 50 kb long, and it's too big for the actual shotgun reads. The actual technique that gives you a consecutive set of characters will give you maybe, on a good day, 500 to 1,000 nucleotides. And you've got to sample this target of 50,000. So the idea was, basically, you take a pure sample of the DNA of interest, say the DNA of Lambda. You run it through a little aerosol mist or a nebulator, and you shear it into a collection of pieces. So the idea is that each strand of DNA, these are all the same copies, each one of those is independently partitioned or broken into pieces. And the important thing is that they're all being broken in different places in different copies. You can roughly control the sizes of the pieces that you get with this process, but you need to basically do a subsequent size selection or gel purification to basically size them so they're all even. So at the end of the day, you have a collection of randomly selected pieces that are length about 1 to 4 kb. Now what you do is you actually insert those into a vector, into a little phage. Transfect that into E. coli, and then get the E. coli to basically produce lots of copies of the vector. The idea basically, this is called cloning, is to get enough copy of one of those pieces of DNA, one of those 1 to 4 kb pieces, that you have enough material to run a sequencing reaction. And then using a primer, typically you can get between 500 and 600 bases. Okay. Now, if you think about that from a mathematical perspective, sorry, I guess it's the mic is pretty directionally sensitive. If you think about that from a mathematical perspective, what's happened is you started out with an unknown piece of DNA, and in that previous slide, if my machine decides to respond, okay, hopefully it's not going to advanced 50. Okay, great. What you've effectively done in the previous slide in the laboratory is randomly sample a collection of fragments from that piece of DNA. It's as if you had gone in and reached in and randomly grabbed out 500 or 600 letters at a time. Okay? And you call that a shotgun data set. In typical technology today, the size of the target might be 100,000 nucleotides long. That's the letter G. Is there a pointer? No. Okay, well, I'll go ahead and work with my pointer, I guess. So you pick about 100,000 base pairs. In a typical protocol, you might sequence, say, about 1,600 reads. And if each are fragments, and if each read is of length approximately 500, then if you think about it, you've collected about a total of 800,000 bases of DNA. Now what's going on there is that, now the target was 100,000 bases, so you've basically way overdone it, right? You would need to really get about 100,000 in order to cover every base once. But because it's a stochastic process and you're reaching in randomly, right, there's a probability that you'll have been unlucky and you won't have grabbed a particular piece. And it turns out that there's a little stochastic theory that says basically that you need to go in quite a few times in order to make sure that with high probability you've sampled every part of that piece of DNA. And typically that's about 8-fold, and we'll talk a little bit more about that. Once you've got that data set, the problem basically from a computational point of view is to build a piece of software that takes those fragments and figures out what you sampled them from. So a classic inverse problem, right? I did something, I grabbed a bunch of data, what was it that I got the data from? And so the idea is to build a piece of assembly software that someone takes all those reads, decides how they go together in kind of overlapping configurations, and at the end of the day can report to you what the original sequences were. Thanks. Okay. The things that make this problem tough are the fact that you don't necessarily know the orientation of the fragments. It turns out that the DNA is double-stranded, and so there's a strand that reads left to right and one that reads from right to left, and you don't know which strand a particular read came from. That's the problem of orientation. It turns out that even though the sequencing instruments are quite good, they tend to make errors. In the old days, it used to be about 5% of the sequence was noise, and in today's technology, this is now around 1 to 0.5%, but you can see all the little red dots represent mistakes, so in the letters. It's the fact that, even though I've gone to 8-fold coverage, or an 8x project, I may not have necessarily sampled everything, so I may not have covered it completely. There may be additional information about how these go together, and we'll see that in a moment. And then, of course, the real fundamental problem is that oftentimes the sequence can contain repetitive elements within it. It's the same sequence that's repeated many times, and those regions can be particularly perplexing from a computational point of view. So basically, by 1990, this technology was going along pretty good, and people had started thinking about doing the human genome actually in around 85, but in 1990, the human genome project was announced. Now, at that time, if this line represents the human genome, this line is extra long. This line here is conceptually 3 billion letters long, so it's much bigger than the 100,000 I was talking about. And to shotgun sequence it, you would have to collect, 70 million reads or so. And people felt at the time, in 1990, that you couldn't possibly write a computer program that could actually reconstruct the genome from a shotgun data set. So what they did is they embarked on a hierarchical plan where they basically developed this method for basically cloning and manipulating pieces of DNA that were about 150,000 letters long. And these are called backs. And the idea was that they would build a library or a collection of these backs, and then they would using a technique called physical mapping, they would figure out how these backs covered the human genome. And once they had done that, they would then pick a minimum set of those, or a minimum tiling, that basically would cover the genome from end to end with as little overlap between the backs as possible. And that would require roughly about 30,000 backs. And then the idea was, a back is only 150 kb, that's not too hard, so let's go ahead and shotgun sequence it, okay, as I showed you in the previous slide. So we have software that can do that. So off we go. Okay, so from a kind of a systems industrial engineering point of view, this process has some negatives, namely it involves both a shotgun sequencing factory and a physical mapping factory. So it involves two separate processes. It turns out that these maps, while conceptually it seems like a nice idea, it turns out that it's extremely hard to make these maps. And in fact, they still can't be made, even today. At least not maps that completely cover the genome. And it turns out that one of the other really big hang-ups is that, well, the actual production of the shotgun read of that 500 or 600 letters, given the clone, is fully automatable in today's protocols. Building that library, actually running it through the aerosol mister, handling the DNA, actually building the clone library, that has to be done by a human being, okay? And that's actually high art, okay? So what that means in this protocol is that it has to be done 30,000 times by some human being. And I get the big one. Are they going to get bigger as the talk goes along? Thank you. Okay, but there was a real plus here, which is that you're spending taxpayer money, okay, and it's every time you shotgun it back, you have new information about the human genome, and it was clear that you were going to get something, okay. So it was low risk, which is good, okay. All right. So in 1995, a bacteria by the name of H. influenza, which was 1.8 million letters long, was shotgun sequenced at Tiger by Craig Venter and his team, Rob Fleischman, Mark Adams, Granger Sutton. And this actually surprised people because it was much too big. The common wisdom was 150 kb, that's about it. That's about, beyond that, the computer starts to do a bad job of things. And a lot of people kind of dismissed it by saying, well, it's a bacteria, it's not very repetitive, it's a very simple genome, it has very nice, kind of stochastic properties, no problem. They got lucky on that one. Okay, but it actually caused me to think about it. Okay, and in fact, I worked with a guy by the name of Jim Weber from the Marshfield Clinic, who's a well-known geneticist. And in 1996, I actually had convinced myself that this was not a fluke, that you really could basically shotgun sequence the human genome, that it was possible to do this. And I wanted to kind of give you a couple of the kind of insights that I had. So the first one was, there was this little theory going around by Eric Lander and Mike Waterman, that basically, I'm afraid you can't see it, but basically G, the parameter G is the size of the genome, then L is the average length of read, and C is the amount of redundancy. So am I going to 8x, 5x? And this little theory said that as a function of these parameters, basically this is how much of the genome I expected to sample. So basically 1 minus E minus C, so you have an exponentially vanishingly small amount of the genome unsampled as the coverage increases. So you see by the time you're up to 10, you're basically at 1. And then this is the number of basically pieces or contigs or gaps, contiguous segments where I haven't managed to sample anything, so the number of gaps. And initially you can see the number of gaps goes up very fast because nothing's overlapping with anything else. And then when you get to about a coverage of 1, in fact exactly 1, this curve reaches its maximum, and then things start to collide. And so the number of kind of contiguous pieces actually, they get bigger, and the number of gaps decreases, and then it basically has an exponentially vanishing tail. And here's the little formula for that. Okay, so let's consider it. So let's take a genome, okay, and the parameters, the length of a read is going to be fixed. You're always going to do 500 bases, right, 600 bases. That's always what you're going to get off the machine. So consider the length of the read to be fixed. Also say that I'm just going to do a certain amount of work. I'm going to do 6-fold coverage. That's how much work I'm going to do, okay? So we'll fix the amount of work. And let's think about what happens in terms of the size of the genome, the target. So let's consider the difference between doing 6x of the back and doing 6x of the human genome, okay? And let's look at certain critical factors. So if you look at this little theory, which says how many contigs am I going to have, okay? It basically says that if you fix L and you fix C, then basically the function is linear in G, right? So the number of pieces you're going to have is proportional to the length of the genome. For back, you're going to have about 4 to 5. For the human genome, which is about 25,000 times bigger than the back, you're going to have about 104,000 pieces. So you're going to have about 104,000 gaps at the end of the day in terms of your coverage, or holes where you haven't sampled it. In terms of coverage, it's 1 minus E to the minus C. That's completely independent of G. You're going to get about 99.75% of the back. And for human, you're going to get about 99.75% of the human genome. If you look at contig length is fixed. Okay, this theory describes some of these other things as well. I haven't given you all the formulas. You get about a 33 kb contig. You get about an 82 base pair gap. If you look at one, for example, the maximum gap, it turns out that the biggest region, unsampled region, is going to be about 150 base pairs in the back. And for human, it's going to be about 2,000. There's going to be a gap where you just didn't sample about 2,000 base pairs. Now, and at first you might think, well, these are different because they grow as the log of the genome size. But it turns out that if I'm going to do 25,000 backs, if I look at what's the biggest gap I'm going to have in any one of 25,000 backs, then it's going to be 2,000 base pairs again. In other words, I apologize, but this is a very simple way of stating that the statistics for 25,000 backs are exactly the same as the statistics for the human genome. Okay? There's no difference, okay, in terms of how much of the genome you've actually covered and sampled. Just to make the point completely clear, if I do a back at 10x, okay, this is what it looks like. If I do the human genome at 10x and I take a back-sized region of it and look at how well that's covered, exactly the same. Okay? There's no difference in the statistics of coverage. Now, to be fair, the problem is that if I do a back, I'm going to get 4 pieces, right? And the problem is then I kind of have to figure out how to, what the order of those 4 pieces are. In the case of the human genome, if I'm lucky, I'm going to get 104,000 of these contigs. But notice I have no way of knowing, like, where they are relative to each other, right? So I have a sack, right, of 104,000 pieces of the human genome, right? I don't have the sequence of the human genome. What you do is you modify the protocol that I showed you the last time. In particular, you use a we're going to take pieces that are a little bit bigger, say about 10 kb. We're going to use what's called a double-stranded vector. So in other words, the DNA before was single-stranded. This vector is actually double-stranded DNA. And the nice thing about that is that I can then start a sequencing reaction from either end of the vector. So the vector has been engineered so I can prime a sequencing reaction from either end, okay? And so I can literally get 500 or 600 base pairs from either end. Now, if I control the length of these pieces so that they're 10,000 letters long, plus or minus maybe 8%, then if I get a pair of reads from both ends, then I know that in any solution I propose, those reads should be about 10 kb apart, right? Okay, to be consistent. Okay. So what Jim and I proposed then, oh dear, I'm afraid you can't see it, is the following protocol for whole genome shotgun sequencing. And what you can't read there is it says collect 6 to 10x in a 5 to 5 to 1 ratio of short, long, and extra long inserts. So the idea is these are the lengths of the pieces that you're putting into the clones, so that I'm going to get pairs of reads that are about 2 kb apart, pairs of reads that are about 10 kb apart, and pairs of reads that are about 50 to 150 kb apart. And I'm going to do that in the following ratio. And for the human genome, to do about 10x, I would collect about 70 million reads organized into pairs. Okay? Now, the critical thing is that by organizing these things into pairs, I can take each one of these little contigs that I'm going to get, and if there are gaps in between them, I'm still going to be able to order them by linking them together with these pairs. In other words, if there's a read in this contig, and a read in this other contiguous assembled region called a contig, then I can actually, if I know that these 2 pairs were sampled from from the ends of an insert, then I know that this contig must precede this one, right? So I can actually link these guys together, and so I kind of draw this big red bus bar to connect them all together, and I call that a scaffold. Now, we did a lot of simulations, okay, to basically determine what these, what these ratios should be, how many of them I should have. And one of the other things that the simulation showed is that these scaffolds would typically be on the order of millions of base pairs long. Okay, they should be like 10 million base pairs long. So what you'll get then from by adding these pairs in is you won't have a sack of 104,000 contigs. What you'll have is you'll have about 300 of these scaffolds. Okay, that are containing basically 1,000 contigs in them. Okay. And they're all linked together, 300 contigs apiece. And these are big enough that you can then use external markers, such as STS markers or other markers or known positions in the genome, to basically pin these scaffolds onto the genome. Okay? The advantage of this process is that it's a single, highly automated process, and it requires only a handful of library constructs. Basically 3, right? So a single human being can do this. And in fact, at Celera, a single human being did do it. It's basically Ham Smith and his assistant, Cindy Fancock, okay, basically built 3 libraries, and they built them with beautiful and exquisite care, okay? Plenty of time for coffee, plenty of time to relax. Ham was under pressure because these libraries had to be very random, but certainly he wasn't having to do it hundreds of times. Of course, the trade-off is that while this is very highly automatable, the problem is that the assembly is much more difficult. And in fact, at the time that Jim and I proposed this, everybody said we were nuts, okay, and that it wasn't doable, okay? And I really liked that because what it told me was that I was onto a good idea. So basically, by the time we advanced to May 98, and at May 98, basically, we're 8 years into the Human Genome Project. 3% of the human genome has been sequenced at this point of time using this back-based hierarchical plan. The physical maps are not complete, okay? You're more than halfway through and you don't even have the physical maps, okay? And the common wisdom was that what we were about to do at Celera was not possible. And it was at that point that Celera was announced. One of the things that changed between 96 and 98, okay, because when I proposed it in 96, it was a bit daunting, was that in 98, a new automated DNA sequencer was coming out that was giving us about a 10-fold throughput increase. One of the problems was that to do a whole genome shotgun of the human genome with the prior technology would have taken, with a great deal of commitment, like 5 or 6 years of hard work to get all the data. And if you think about this shotgun protocol, you don't have anything, right, until you've collected all the data, right? So you have to wait 5 or 6 years, and then you're going to find out whether or not it works or not, okay? Way too daunting for anybody. Even I conceded that was a little scary. But I was trying to get everybody to do a pilot, right? I was cruising around Europe, right, trying to get people to do some, a fungus or something at 30 megabase pairs, right? To try something, okay? Proof of concept, okay? But it was really a hard slog. All of a sudden you have this machine that means that in a single operation you could probably do all the sequencing in 9 months. Psychological barrier has suddenly gone way down, although the spend is still pretty darn big, okay, before you know whether you got anything, okay. So that was one thing, okay, was we knew that we could build this factory. The other one was that, of course, this whole genome shotgun sequencing strategy looked like we could really automate things. And another really lucky thing was that 64-bit computing was just coming in. So the computers were just now we were able to actually think about doing this compute in memory, okay, on a machine without the development burden of having to think about 32-bit. The genome is 3 billion, 2 to the 32 is 2 billion, right? So you can't model the thing in a 32-bit machine. And in fact, we built this huge Ganga Supercomputer center. It was kind of fun. It was at one point one of the most powerful civilian supercomputers, but things moved so fast that the numbers in 1999 were 1.5 teraflops with 100 terabytes of storage. And that was big at the time. We were pretty proud of that. And we built this huge sequencing factory floor. And you have to remember, when I arrived at Celera in 1998, in August of 98, I arrived in a big building. There were 19 other people, and we had $300 million in cash, okay? So that's what we started with in August of 98. Keep that in mind, okay? So by basically September of the next year, 13 months later, we had a sequencing factory built, okay? We were up to about, 300 employees, and we actually sequenced the genome of Drosophila, which is 120 million base pairs long. So Drosophila is about 1/30th the size of human. It was our 1/30th scale model. So that was our test, right? Okay, even Craig, as flamboyant as he is, okay, wanted to make sure that we could at least pull it off on the 1/30th scale model. And I was mighty pleased too. I would have wanted to have gone directly to the human genome. But basically, and we published this in Science in February, so basically a year and a half later. In March, Celera and the Human Genome Project basically declared a tie and published their human results. And I think that where the real power of whole genome shotgun sequencing shows up is the fact that we were accelerating. Because literally 4 months later, we had sequenced the mouse genome. Okay, all right. And that was another year before the public effort got done. And you may have noticed that they used a shotgun approach. Okay, but they were playing catch up. And in fact, the DOE who had been quite friendly with us, had noted what we were doing and actually did the fugu fish in October of 01. And December of 01, we had actually sequenced already the mosquito. So we were running a full line pipeline. And today, I think it's fair to say that this has become the paradigm that everyone is using. How do you actually do this thing? Okay, I'll talk a little bit about that. And I like this cartoon because it kind of shows that it was the idea of actually what to do with these 70 million reads. It actually turns out I had 40 million. What do you do with all of those reads? And the other thing that I like about it is that this guy here is saying, I think I found a corner piece. And in some sense, he's got the zen of it exactly right, okay? Which is that you basically do the high relief parts of the puzzle first. In the case of building a whole genome assembler, the idea is to basically figure out which portions of the genome are unique, are non-repetitive DNA, and to put those together first, okay? It's like the high relief and the corner pieces in the jigsaw puzzle. It's exactly the right analogy, okay? So just to give you an idea of the scale of the data set, we had 27 million reads that we had produced at Celera. Another thing that I want to mention is that we didn't have paired data for everything, but we had the 2, 10, and 50 kb pairs. And one thing I want to point out, though, is that a pair is not always a pair. So it turned out that 1.4% of the time a particular pair could not be a pair. There were basically tracking problems in the laboratory, and even though we barcoded everything and we were as careful as we could be, it was still sometimes the case that this pairing relationship, the fact that you came from one end of the insert and from the other, got lost. So you can't completely believe that. It's not a completely true piece of information. The other thing that I want to emphasize, and that I think goes unnoticed, is that we actually sequenced 5 people, okay, different people in different degrees, which meant that not only did we get the sequence of the human genome, but we also got a tremendous amount of information about the polymorphisms within the human genome. In other words, the genetic variation that makes us different, okay? So we had a SNP map that you could use for high-density marker studies to basically track down sources of diseases. So the design of this assembler was basically to detect the repeats and so avoid being misled by them. That was the essential idea. It was also to use these mate pairs in a strong way that had not previously been done. And the other thing that in thinking about how to build this is that I really wanted to make sure that I had a strategy that would make moves that were almost certain to be correct. In other words, my sense of it was that we were going to be highly scrutinized, and if we put out something that was too aggressive and had a lot of mistakes in it, we were going to be crucified, okay? So I thought, whatever I put out there, it's got to be just right on the money, okay? Because it's going to be looked at. And it's actually, that pressure actually led to an interesting design, I think, in many ways, that exterior political pressure. And so one of the things I think that a lot of people aren't used to is that we don't feel obligated to use every piece, okay? So we're willing to leave parts of the jigsaw puzzle unsolved, okay, if we can't solve them perfectly, okay? All right, so I'm going to just briefly kind of tell you how I turned this trick, okay? Actually, I want to do some other stuff later on, so I think I'm going to just skip this stuff and go, I have a very condensed version of it, and I'm just going to do that. Okay, I hope you don't mind. Be glad to talk to you about it later. Suppose that this is a genome. Okay, this is the basic recipe. Okay, so suppose this represents a segment of the human genome, and suppose that these red things here represent DNA sequence that's repetitive. I mean, it's some motif X, and suppose it's exactly the same motif over and over again. Okay, the idea basically, and I won't tell you, I've skipped over exactly how I do this, but basically what we did is we worked out a cute trick for figuring out which portions of the genome, which of the reads came from portions of the genome that were not repetitive, okay? Because the problem with the repetitive stuff is it looks like it all goes together, right? And, it looks like it's all green, okay? It looks like it's all C pieces, okay? And there's no relief to tell, which C piece goes with which C piece. And if you do that, you end up with mush, right? You connect things in the wrong order. But in the high relief parts of the puzzle, where the DNA is relatively unique, it's easy to tell who belongs with who, and there's no problem figuring it out. So that was the key trick, and you can read the technical papers to do it. But the idea was basically I'd figure out what the unique parts. Now the repetitive parts typically aren't that big. They tend to be at the biggest, they tend to be about 5,000 letters long. And so if you use these paired reads, and you use pairs of them, you can link them together. And if you just ask that there be at least 2 of them, it turns out that the chances that 2 of them agree with each other and are both wrong is extremely rare, right? Because I said 1% of them could be wrong. The chances that they're both wrong in the same way is extremely rare. So if you just ask for doubletons, okay, you can connect things together into these scaffolds that are highly reliable. Okay, and then it turns out that you can even get at the interior of the repetitive reads, segments, by basically using reads where one read is in the unique DNA, and by virtue of knowing this distance, the distance from here to there, this other read is placed inside this particular repeat hole. Okay? Now, what I do is I basically look at all of the reads that by virtue of their mates, the reads at the other ends of the inserts, have to be inside this hole, and then I assemble them. because that's a single repeat all by itself is not repetitive, right? It's only repetitive in the context of everything else. But if I put the right reads in the right hole, then assembling them is no problem. Okay, and I can put them together. And you can ask, and so we did this in 2001, and then ensued a huge debate about how good it was. Okay, I don't know if you heard any of that. Okay, but we were all going at it. Okay, and everybody didn't like Craig and wanted to show that that Craig hadn't done it, and so all the public guys were going after him, and basically the problem is how do you prove something is right when you have nothing to compare it to? Okay, I mean, okay. Now, we did have the public version, okay, and we did do the comparison, but that was another whole set of algorithms, okay, and it took 6 months, okay, for us to write the code and to actually do the compute, right, because you're talking about 2-3 billion base pair strings with all the caveats and stuff, okay, and I won't bother you with what we found out, okay. I just want to because it's good for us and bad for them. So I'll just state that and leave it at that. But you can look at some things. You can look at kind of your statistics of how many pieces you have, how big they are, how many of the gaps they are. Well, I don't know if that means anything, right? I mean, I can get a computer program that sticks everything together and it can be completely wrong, okay? But the stats will look really good, okay? So that's not so great, okay? Another thing you can do is you could take these paired reads, right, and see, if they're all, at the appropriate distance from each other. But of course, we can't do that on our own stuff because we used it, right, to solve the problem. So we should be consistent with it. So that doesn't tell us much, okay? Of course, we used it to look at the public stuff, okay, which again told us some interesting things, okay? You can, now it's getting a little bit better, but what you can do is okay, so nobody else has sequenced the human genome, it is the case that people have built high-density marker maps, okay, STS marker maps, okay? You can compare yourself to those, okay? We did that. Within the level of accuracy of those maps, we looked great, okay? Finally, recently, I've been able to actually do what I've always wanted to do, right? Which is this December, the sequence of Drosophila was finally finished. I mean, a group had gone through, basically vetted everything, done double-ended sequencing, checked everything, mapped it. I mean, basically, the U-chromatin of Drosophila, the 116 megabase paired U-chromatin of Drosophila is finished and checked off by human beings, okay, in an independent effort. And so what was really cool is that we compared our assemblies, okay, of Drosophila against that. And that's really the gold standard, right? How close did we get to a real target? So, we had the typical shotgun input. One of the things that we did is we compared 3 different versions of the assembly. We compared WGS, we compared basically our technology at different points of time. So WGS1 basically represents the assembler that we had at the time we reported the first sequence of Drosophila. We made a number of improvements, and then WGS2 is kind of a politically important milestone because it's the assembler we had at the time we published the human genome, okay? And then WGS3 is kind of the last one we did on melanogaster before I left Celera, so it represented kind of our best assembler at the time. Because you have to remember, this assembler, right, is not, building code is not, for those of you that aren't software scientists, I mean, building code takes a while. I mean, it takes years to get a really robust piece of software you really kind of, especially with a problem this big. Okay, so we are continually refining things and getting better and better and better. State of the art moves forward, right? So you'll see progressive improvements as you go through this. And there's still more improvements that can be made. I mean, I don't think that WGS3 was the end of the last word. But so you can look at the statistics. And again, I really point this one out just to kind of show you that the statistics are really deceiving. Like, if you look at WGS1, it had 816 scaffolds, and WGS3 had 2,775, and you go, geez, it's getting worse, okay? All right? It's not getting worse, though, because, look, with the first assembler, we reconstructed. Remember, we only report what we think we're sure of, right? So we were sure of 122 megabase pairs of sequence. But by the time you get to WGS3, we're giving you 137 megabase pairs. So we're giving you 18 more megabase pairs, okay? So you actually have to go in and look at the assembly. If you look, inside of these 2,775 scaffolds are scaffolds that completely contain those 816, and then a whole bunch of smaller ones that account for the rest of it. In particular, if you look at a genome, okay, typically you look at a chromosome, any chromosome basically has a centromere, a central heterochromatic structure, and then some telomeres. These regions are very, very repetitive. I mean, in fact, they basically migrate into stuff that's alpha satellite, that's basically a triplet, repeated, a bazillion times, okay? So it's impossibly repetitive in here. So an intrinsic assumption of any sequencing project is that you're sequencing what's called the euchromatin, okay? The stuff that doesn't stain too dark, okay? All right, if you're a cytogeneticist, okay? And so you're basically going after the sweet spot, this yellow part, okay? And this is true of all of the projects because you really, in some sense, can't sequence that. And so the question is so kind of the idea is that WGS1, in some sense, nicely covered the euchromatin, but as we improved the technology, we were actually beginning to reconstruct stuff inside the heterochromatin. So, the question is if you look at the numbers, this assembly consists of 2 scaffolds, and this one consists of 6. Which one would you rather have? Right? I mean, you want the second one, right? Okay. Because it's got useful information. Okay. So, in fact, if you compare now to just the euchromatin, just the sweet spot that was technically finished, so release 3 contained 116.91 megabase pairs of euchromatin, you can see that basically our assemblies, basically even the very first one, completely covered it. And in fact, we reconstruct almost all of the sequence, okay, with very, very few gaps and very small gaps. And in fact, I'll point at a couple of numbers. We basically covered 99.91% of it, okay, and in terms of sequence, if you subtract out the gaps, we had 98.93%. So right out of the assembler, we basically reconstructed 99% of the euchromatin, okay, right with a shotgun technique. In addition to that, though, there's another 20.7 megabase pairs in that assembly that belongs in the heterochromatin, okay, which is something you won't get with a back-based project. Okay, you can't get this using backs, okay, because it doesn't clone in the back. Okay, and it contains basically, 266 newly predicted genes. They're probably interesting. Okay. Okay. In terms of, how good was it in terms of the reconstruction? The initial assembler had some problem with repeats. But by the time you get to our latest technology, what you're seeing is that inside of that assembly, there were basically no errors, large errors in order and orientation. There were 3 local errors where 2 little contigs were switched. They were in the wrong order. And the total amount of sequence involved was 5.5 kb. Okay, so there were little teeny contigs that just weren't quite right in the right order. And we had one repeat reconstruction that was a chimer that had basically a bit of the wrong repeat in it. So basically, we made 4 mistakes across 120 megabase pairs, okay, in the computer. If you actually go down and you actually look at the sequence, so you go say, okay, what's the quality of every individual letter, okay? The standard is to have basically 1 error for every 10,000 base pairs, okay? And if you look at this over all the sequence, basically by the time we get to WGS3, without any human vetting, we're down at basically the standard, 1.1. Now this is a little bit unfair though, because it turns out that repeated constructs are not nearly as high quality as unique sequence. If you look at just the unique sequence, which is basically more than 90% of the genome, it's actually 0.4 errors, okay, per 10 kb. And you can see that the interspersed repeats, it's basically 1 in 1,000. And for the tandem repeats, which are particularly difficult, it's 0.5%. So what I would say with whole genome shotgun sequencing is that it's perfectly reliable. You don't need to do a thing. Just take it right out of the box for unique sequence. But for your tandem repeats and your interspersed repeats, if you're really planning on doing evolutionary studies on those, you probably want to do some additional finishing work. Let me talk a little bit about what I think is coming down the pike, okay, in terms of sequencing. So I think there are basically 3 new technologies. So you've heard about this idea about the $10,000 genome, right? So how might that happen? So there's, you can't see it. There's solid-state sequencing in picowells, okay? This is really kind of cool stuff. They figured out how to make these really using kind of fiber optic stuff. They figured out how to make these little teeny wells that are 40 angstrom across or nanometers across, and basically they can affix reagents on some beads, and they can actually do pyrosequencing inside these wells. So they can run an individual sequencing reaction inside these wells. There's a company that's doing this. I think they're going to be operational next year. Okay. Now the problem, and these instruments will basically produce 100 megabase pairs per instrument in 50 base pair reads. Okay. Where you can get pairing information that's anywhere between 300 base pairs and 1 KB apart. So the idea is that this is compared to basically 1 to 2 megabase pairs per instrument per day today, with about 800 base pairs. So it's basically a 50-fold increase in sequencing output, total base pairs. Unfortunately, you get your base pairs really short. You get 50 base pairs, okay, instead of 500, and that makes assembly really hard, okay. Because now you can't get through even, an ALU element, okay? So what we're thinking is that that's probably going to be good for resequencing efforts and for doing backdrafts. So if you want to actually sequence backs, though, which is 150 KB, and I've done some simulations, okay, it looks to me like with a good enough assembly, we should be able to actually do de novo assembly of backs. Okay, so if we want to go with a back-based approach, this might offer a significant reduction in cost, because this is also cheap too compared to the other methods. Okay, but I can't talk about the numbers. Another idea that I like, which is a little further away, is detecting the incorporation of DNTP into a fixed Pol II complex. So the idea is this is again nanotechnology. You make a little nanowell. You isolate and affix a Pol II complex in that nanowell. Okay. Now the nice thing about polymerase is that basically it's the molecule that makes a copy of a piece of DNA, and you can watch it incorporating the bases while it's making the copy. Okay. And the nice thing is it's very processive. It's kind of chunking along at a nice kind of even jog. And every millisecond or so, a new DNTP is coming in, okay? So the beautiful thing is that even though it's a single molecule, you know where to look inside the well, and you know when to look every millisecond or so, okay? Which means that you actually can do single molecule detection, okay, with current, state-of-the-art optics, okay? So what I've seen here is that I've actually seen signal. Okay, people are actually getting a signal. Okay, but it's not in a commercial format. Okay, and it's still kind of an experimental prototype stage. The capillary gel stuff that we used at Celera 98 was basically in this state in 1988. So if I take that lesson, I would say that in 5 to 10 years, we will have instruments based on this technology. They're going to produce huge reads. They're going to produce 100,000 base pair reads. Okay, so now it's going to go the other way. Instead of them being 50 base pairs long, they're going to be 100,000 base pairs long. But they're going to be really noisy. Okay, so the error rate, I better get over here. The error rate is going to be, could be as high as 20 to 30%. So now the problem becomes, okay, I give you Ganga reads, okay, but I give you a lot of error, okay. Give me the sequence of the genome, okay. Finally, I'll talk about Nanopore, and I know it's been very hyped, but in my opinion, as a sequencing technology, as opposed to a SNP detection technology, okay, I'm not convinced that it's going to be there anytime soon. You can detect the fact that a piece of DNA is moving through a nanopore, but you can't actually see the individual bases, okay? I mean, if you've looked at the signal, it's noise, okay? It goes through like that, okay? All right, you can't control it. You can't slow it down, okay? It's there and gone. All you can tell is that it went through, okay? So I haven't seen a signal. So kind of my rule of thumb is until I've seen a signal, I won't predict when it's going to be a commercial instrument, okay? And, somebody might make a breakthrough in the materials and stuff. Might get a signal next week. Okay, but until that happens, I make no prediction. Okay, I think it'll eventually work, but I have no prediction for that. Okay. All right. May I take just a couple more minutes? I want to kind of talk about so I've done the sequencing of the human genome and a few other things. Kind of been there, done that. Okay. I've been wondering what to do next. Okay, and where we're going next, and so I've kind of picked as my own target and the thing that I'm interested in, really trying to understand development. So here's a little movie of the first 12 hours of development of Drosophila larvae. And I want to know why that's happening. okay, and how that's happening. I want to know what the program is, the genetic program. So inside the DNA of that Drosophila, inside that 116 megabase pairs of euchromatin, right, are all the genes and the regulatory signals to make that happen, okay? But we don't know what it is, right? We only know part of the code, and we really don't know how it's operating. So the question is how is that happening? Hopefully I can go to the next slide. So the goal of molecular biology has always been basically to understand cells and organisms as molecular systems. I think what's really getting exciting is that, I think that we really see that we're going to be able to get there, that we're really at some point going to be able to think of a cell as a collection of particles, proteins and other molecules that have particular properties, and we'll have a description of all of those properties and their interactions, localizations, and where they're doing things and when they're doing it. And while that may not be necessarily good enough to do, simulations that are predictive, it will certainly be intellectually a fascinating data set and one that will lead, I think, to a tremendous amount of discovery and correct intervention. In particular, I think that we'll be able to pick a target, pick a protein, and really have a chance of understanding what knocking it out means, okay? All right, in the context of the entire system. I think that we'll also be able to predictably start to modify these systems, and of course these have tremendous implications for our well-being. But in the meantime, we've got to do that, and I don't think that human beings are the right thing to focus on, okay? Because these questions are too fundamental, and the human model has too many complexities, okay, associated with it for us to start. This was a very general, I was told this was a general audience, and so just in case, the basic building blocks are DNA, which contains the store, RNA, which basically converts the DNA into information, proteins, which are in some sense the executors, and small metabolites, which are basically the fuel and the inputs to this system. So basically, if you have a genome, the idea is that within that genome are regions that encode for genes. These genes are transcribed into this messenger RNA in anticipation of being turned into proteins. These mRNA are oftentimes spliced, so they're actually edited, okay? This is all taking place within the nucleus in the case of eukaryotic cells. This mRNA is then transported outside the nucleus, where in the cytoplasm it is then translated into a protein. So in other words, there's a section of that genome that's coding for protein. Okay? The system is recursive in that this entire process that I just described is itself mediated by proteins, by polymerase, which is a protein complex, the ribosome, which is a protein RNA complex, and so on. Oftentimes these proteins then need to be clipped by other proteins to be activated, where they then serve in metabolic roles in terms of synthesizing molecules, degrading old molecules, and creating energy to make the whole system run. A particularly interesting class of proteins are those that are actually secreted, that are messengers, that actually leave the cell through a pore, then bind to a receptor on the surface of another cell, initiating a cascade, a signaling cascade, in which, for example, a phosphate group might be exchanged across a collection of proteins, and ultimately phosphorylate a transcription binding factor, which is a protein which controls the expression of a gene. Okay, all right, so I just closed the loop, okay? So that's biology 101 in like 1 minute, okay? So that's the system, okay, that we're trying to understand, okay? So to try to put names on parts of that, okay, first is getting the DNA sequence. That's called sequencing. Second is figuring out what's actually in the genome. Where are all these proteins encoded? Where are the little binding sites for the transcription binding factors that are turning things on and off? That's called annotation. Once you've got all of the parts, you want to know how they interact with each other. Those are the pathways. When they're interacting with each other, you'd like to know what they're doing. That's their function. And, of course, it's very important to know where. Okay, subcellular localization. So there's some terms for you. Okay, and so what I'm very interested in is executing the following proposal based on Drosophila, and I'll tell you why Drosophila in a minute. So the idea is to basically sequence 10 to 20 species of Drosophila. So I don't want just one. I want 20 related species of Drosophila. What I'm going to then do is do comparative genomics in order to understand, to basically, first of all, answer interesting questions about the evolution of these genomes, okay? I mean, we already have very interesting pictures that show that in the short term, there's a lot of recombination and duplication and translocation, and it all seems to be mediated by the repetitive elements, and it seems to be happening near the centromeres. This seems to be the melting pot, okay, for evolution. But the more important thing for me, actually, and the real reason I'm interested in doing this, okay, the primary reason, is to accurately annotate the genes and the regulatory signals. So the idea is that rather than doing a lot of in vitro and in vivo experiments to try to understand whether or not I've captured a particular protein in the genome, by looking for conservation of signal, okay, conservation of sequence, essentially. Well, it's a little more complicated than that. I should be able to accurately divine where all the genes and regulatory signals are. But you don't stop there. Once you've got an annotation, you do need ultimately to verify that annotation. And furthermore, I would stipulate that it would be very exciting to basically capture all of the transcripts and gateway clones to facilitate basically experimentation. Why should everybody be doing this? We should make one resource, get all 14,000 Drosophila genomes, get them trapped into gateway clones and distribute them. Have a commercial entity distribute them for them. Once you've figured out what all the parts are, then you should build expression assays. Okay, not before. Then you should build highly tuned expression assays and apply them to survey the things. And then I further want to utilize mass spec to basically systematically explore these signaling cascades that I've been talking about. The state of the art will allow that. The particular reason I chose Drosophila is that it's an interesting model. It's a metazoan. It has development, differentiation, and behavior. So it has a lot of the things that are important. Bacteria are just, they just don't have enough phenotype for me, okay? They're just, whereas here you've got clear phenotype that you can associate with the genome. It's a nice lab animal. It's easy to stock. There are lots of experimental techniques for it. The other thing is that it's the 1/30th scale model, okay? So some papers were written about how to compare the human and mouse genomes, okay? These things are 30 times bigger. Just getting the computes off the ground, okay, is a nightmare, okay, even with today's machines, okay? Not everybody can do it, okay, so not every computational investigator can do it because they don't have big enough computers. Not that many people want to chew that off, okay? The people wrote heuristics. They cut corners all over the place to try to reduce compute time because compute time was the issue. Science wasn't the issue, compute time was the issue. And you can see they got different results. Nobody knows why the results are different. You can't go check anything, right? Experimenting on mice and humans is expensive. With Drosophila, it's 1/30th the scale, okay? Which means that the computes are a lot easier to do, okay? Means that basically everybody in this room is computational scientists can take that data set and do the compares, right? So instead of just, a few special guys in the big computer club being able to do this, right? We'll all be able to do this. We'll be able to do better comparative genomics, right? We'll get better comparative genomics tools, right? So I think this is a really this is the right level at which to be doing the science right now, okay? In terms of doing comparative genomics. Develop the tools on the 1/30th scale model. Once you know they work, then let's scale them up. Then let's do the big gun with compute on human and mouse and all the other barnyard animals, okay? But doing and the other thing is that there's a huge community. Another reason that I chose Drosophila is that it's a great model for human, and it has a large community. So that anything that we do as computational experts, it's going to be vetted by the biologists. There are going to be a lot of eyeballs, okay, expert eyeballs, okay, looking at what we're doing, right, telling us whether or not we're right or not, okay. And they're also going to, and it's real easy to go in and do an experiment, okay. If you want to check something out, to do a genetic cross or whatever it is that needs to be done to get it done. And it is a good model for the human. It's not as complicated as human, but basically my thesis and my sense of it is that every kind of control circuit, every paradigm for control, okay, of development and differentiation that you would find in a human being is probably in there in Drosophila, right? Not in the same copy number, maybe not in the same multiplicity or the redundancy, but all the paradigms are there, okay? So I think this is a great model, and it's a much more amenable model. So this is the mouse human. So one of the things that we're actually putting together is a white paper to actually do this particular ladder of Drosophila sequences. They range from basically 2 million years apart out to about 90 million years. 90 million years may not seem like very much to you, but you have to remember that Drosophila breed, in terms of generations, it's a lot more generations of flies than it is of humans, okay? Anyway, I won't bore you with how we do the comparative techniques. I will say that there are a lot of other kind of interesting data sets to think about. So, you know, and there are a lot of genes that we don't know about. So here's this gene, CG14897. So we know a lot about this gene, okay, meaning its number, okay, all right? But you take that gene, you build a little in situ probe, and you actually just watch it in a confocal microscope, and you look at where it's actually being expressed, spatially as well as temporally. And you watch it, and basically in stage 1, it's kind of all over the place, okay, at the very early stages. In stage 4, it does a weird thing. It goes to the pole, so it has something to do with orientation. But then in stage 8, its role becomes clear. It's actually associating with the neuroderm, okay? Stage 11, it's the protocerebrum and the primordium. And then finally in stage 14, you can see clearly that it has something to do with the central nervous system. Okay? So, why that particular pattern? Okay? And I'm really interested in these things because a lot of people are basically looking at, you know, expression arrays. They're kind of looking at like the level, but it's the level averaged over the whole organism, right? So I can tell you that this gene, if you look at its expression averaged over the whole genome, it goes like this, okay? All right, it doesn't tell you much, right? Look at that picture. It's got space and time, okay? It tells you a lot more. There aren't that many other genes that are expressed that way, right? So the whole idea is what can we, so I'm very kind of interested in trying to do things computational with these images. And the goal eventually is to come up with some kind of circuit diagram for transcriptional control, for the development and the turning on of these things. So this is stolen from Davidson's paper on the sea urchin, where he's basically taken 50 of these transcription factors and kind of built a circuit. I mean, it's not clear that this is right, okay, but a lot of people are really captivated by the idea that we're going to be able to build these models, okay? And I really do think that it's happening. I think that in the next 5 years or so, we're going to actually begin to really understand how this unfolding is taking place, to really begin to build models of it. And I'm very excited about that. Okay, so I hope you enjoyed that. Of course, I did all of this by myself. Actually not. Okay, I worked with a great crew, and this was the seed crew that really, I think, was a wonderful group that did that built the original assembler for Drosophila, which was really the beginning of it all. Okay, thanks. That's it.
You suggested that the original public draft of the human genome had some problems, which I think everybody knows. But Celera promised to release their assembly, and they didn't. Now you're suggesting that are you suggesting that the latest assembly has big problems too?
No. The finished assembly is fine. The thing that they've just announced is fine. It's been improving and improving. So I've never been clear about this, whether or not it was released or not, the Celera assembly. It was available on a DVD, and anybody who wrote in for it was sent one. I think that it was available. I mean, you could get it.
I was under the impression that in order to get it, you had to sign all kinds of waivers.
You had to sign a contract that said you wouldn't do commercial work with it, right? And I don't know what the other strictures were, but you could get it. You could get it and do science on it. And it was archived with the people at Science. I mean, so it's not like but yeah, I don't know. I mean, we made a DVD. I know that we distributed it to at least 500 individuals. So people did get it. People were willing to sign the clause. I think the problem was that there was a stricture that you couldn't use it to write global bioinformatics paper, and so all the people that wanted to do global analyses didn't want to go near it, and that was a stricture. That was unfortunate. But it was available. I mean, you could look at it. It wasn't like it wasn't verifiable. And the other question was, there's no problems. No. So there were problems with what they produced in 2001. In the last 2 years, they've done a tremendous amount of finishing work and resequencing of backs. And as they've done that work, it's gotten progressively better and better to the point where today it's extremely good. It's very reliable. Did I answer your question?
Yeah.
So there's another technology, microarray
I can't hear you. I don't hear very well. Sorry.
There's technology called microarray.
Yes.
And in your opinion, what do we expect from the results of microarray? What kind of results do we expect?
I guess the question is what can you get from microarrays? So microarrays are very popular. There's been a tremendous amount of work on them. I think they have uses, but I'm not a big fan. Okay. The uses that they've been good for. So basically, the idea of a microarray, in case you don't know, is basically you put down little bits of DNA, pure DNA, on spots. You do it in a very high density way, and then the idea is that there's DNA that matches the DNA at the spot. It lights up, so you can figure out kind of how much of each DNA there is. And so it's been very useful for, say, for example, typing cancers. So if you kind of think of the intensities as kind of a Rorschach plot, okay, or as a pattern, you can tell the difference between cancer A and cancer B, okay. So it's really useful for understanding. It gives you a pattern, an impression of the expression that's significant enough to categorize cancer A from cancer B. But if you want to know, if this gene goes down, does this gene go up? And how much does it go up by? It's much more difficult to get that kind of information from it, okay? And unfortunately, it's just too noisy right now. The signal is too noisy to do that. But there are some technological advances that are coming down the pike. And the next generation arrays, where the signal-to-noise ratio is improved by about a factor of 10, may really begin to become useful for kind of looking at and dissecting pathways. So arrays have not been really useful for looking at and constructing individual pathway and expression dependency information. Did I answer your question? Yeah. Yeah.
I have a question. I've learned that you're related with the most popular algorithms for searching the sequence, which is called BLAST.
Yes.
BLAST, right now, currently, we have two versions of BLAST. That is NCBI BLAST and whole-BLAST. Can you compare the characteristic and make it?
No, I can't. I can't. I basically conceived of the basic algorithmic idea for BLAST back in 1989, and was on the team of 5 that built the 1990 version. I haven't had anything to do with it since. So I mean, not other than use it occasionally. So a lot of people have been obviously following up on that, and there are now. I mean, there's BLAST Z, there's Mega BLAST. I'm getting confused by it all at this point. I think they all work about the same.
Okay, well, thank you again.
Okay, thanks.